Can we reset the age of a cell? In 2006, Japanese scientist Shinya Yamanaka did something once thought impossible. He took an old specialized skin cell and reset it into a stem cell, a cell with no age, no identity, and unlimited potential. The clock didn't slow down, it reset. The discovery raised a profound question. If a cell's age can be erased, what is aging actually measuring? That question would reshape modern aging research and change how scientists understand the process itself. Your DNA contains roughly 3 billion base pairs, the code behind every protein, structure, and function in your body. And that code barely changes throughout your life. You die with almost the same DNA you were born with. So why does an 80-year-old cell behave so differently from a newborn one? The answer lies not in the DNA itself, but in the instructions that control it. Wrapped around your genetic code is the epigenome, a layer of chemical signals that tells each gene when to activate and when to stay silent. A liver cell and a brain cell contain the same DNA. Their difference comes from which instructions are being read. Throughout life, the epigenome constantly adjusts to stress, damage, and the environment. When we are young, those instructions are precise. The right genes are active in the right cells at the right time. But over decades, the system becomes noisy. Instructions become corrupted. Cells begin to lose their identity, not because their DNA is broken, but because the information controlling it becomes unclear. This is David Sinclair's information theory of aging. Aging may not be hardware failure, but software corruption. The code is still there. The problem is that the system struggles to read it correctly. And if the original information still exists, then perhaps aging is not only about damage, but it may also be about forgetting. That is exactly what Yamanaka's discovery revealed. When he introduced 4 genes, known as the Yamanaka factors, into an adult cell, something extraordinary happened. The epigenome didn't simply change, it reset. Decades of accumulated noise and biological memory were erased, returning the cell to a state similar to early development. But there was a problem. A fully reset cell is not a younger version of itself. It is a blank slate with no identity. In a living body, that creates a dangerous possibility, a cell that has lost both its age and its purpose. For years, this seemed like an impossible barrier. Scientists could make cells young, but only by removing the very instructions that made them useful. Then came a new idea. What if you didn't need a complete reset? What if you could briefly activate the reprogramming process, just long enough to clear the damage, but not long enough to erase the cell's identity? This became partial reprogramming, not a full restart, but a targeted repair, removing the noise while preserving what the cell was designed to be. In 2020, Sinclair's lab tested whether partial reprogramming could restore aging cells without erasing their identity. Using 3 Yamanaka factors, delivered into the eyes of old mice, they briefly reset the cell's epigenetic state. The results were remarkable. The retinal cells did not grow back, and the DNA itself did not change. But the cell's instructions shifted toward a younger state, and vision improved, sometimes returning close to youthful levels. The cells were not replaced. They were not repaired in the traditional sense. They were reminded. The information needed to function like younger cells was still there, hidden beneath years of biological noise. The experiment suggested something profound. Aging may not be the loss of information, but the loss of access to it. This discovery raised a new possibility. If aging leaves measurable marks on the epigenome, then perhaps we can read those marks and determine a cell's true biological age. Steve Horvath at UCLA spent years mapping these patterns. By analyzing chemical tags across hundreds of locations in the epigenome, he created the epigenetic clock, a tool that can estimate how old a cell is biologically, not just how many years a person has lived. The results revealed something surprising. Biological age and chronological age are not the same. A 50-year-old can have cells that appear much younger or much older. This difference predicts health, disease risk, and lifespan. Even more fascinating, Interventions linked to healthier aging also appear to slow the clock. It isn't just a measurement, it is a window into the aging process itself. Here is the honest part. Partial reprogramming is still experimental. It has been tested in cells, tissues, and animals, but not safely in a whole human body. The Yamanaka factors are powerful, and using them incorrectly could create serious risks. The difference between restoring youth and losing cellular identity is still a delicate line. The science is moving quickly, but the gap between a mouse experiment and a human therapy remains enormous. What has changed is not that we have solved aging. What has changed is how we understand it. For most of history, aging was viewed as inevitable damage, a system slowly breaking down. The epigenetic evidence suggests something different, that much of aging may be a failure of information, not a loss of the code itself. The instructions are still there. The cell hasn't forgotten everything. It has simply lost the ability to read them clearly. And that is a very different problem, one that may have a very different solution. We began with a cell that could be reset, and discovered that aging may be less about damage than lost information. The evidence suggests cells still carry a younger version of themselves. The question is no longer if it exists, but whether we can safely restore it in a complete living system. The answer is being tested now. One mechanism understood, many questions remain. And the biggest question is not whether we can make cells young, but what happens when we try to change the entire system.